मैं राजवीर सिंह शेरान एसोसिएट प्रोफेसर इन केमिस्ट्री SRR मोररका गवर्नमेंट PG कॉलेज झुंझुनू। फर्स्ट ऑफ ऑल गुड आफ्टरनून टू ऑल दी स्टूडेंट्स ऑफ MSc फाइनल। पिछले वीडियो लेक्चर में वी हैव डिस्कस्ड अबाउट द प्रोटीन्स, यूनिट सेल, biological cells. Today we will discuss the structure of protein and bonding in proteins. So first of all we discuss bonds related to protein structure. The protein structures are confirmed by strong bonds, peptide and disulfide bonds, and secondly weak bonds, hydrogen and hydrophobic bonds. So first we discuss strong bonds. Peptide bonds, the compounds formed by the condensation of two or more or same or different amino acids are called peptides. During the formation of peptide, the amino NH2 group of one alpha amino acid and carboxylic acid group of one another molecule of the same or different alpha amino acid get condensed with the elimination of a water molecule. During the condensation, a bond of the type CONH is formed between the two amino acids. The amide linkage is CONH is known as peptide linkage or peptide bond, as shown in the figure with arrow. Peptide bond, alpha amino acid first plus second alpha amino acid, these two combines with the elimination of H2O and they form peptide bond. The peptide formed due to condensation of only two molecules of the same or different amino acids is known as dipeptide. Similarly, the peptide found due to the condensation of 3, 4 or large number of molecules of amino acid is known as tripeptide, tetrapeptide and polypeptide respectively, as shown in the following reactions. All peptides contains a free NH2 group at one end and a free carboxylic acid group at the other end. These are called AND groups or terminal groups. Some biologically important peptides are angiotensins, octapeptide, nonapeptide, oxytocin, nonapeptide, etc. The primary structure of protein is derived from the peptide bonds, which can be established by the following evidences. Proteose produces polypeptide by hydrolyzing proteins. They can also hydrolyze peptide bonds. The proteins, insulin, etc. is synthesized by linking amino acid by peptide bond. Polypeptide bond link proteins reacts with the biuret reagents which is suggestive of two or more peptide bonds. Then disulfide bond. The disulfide bond may interconnect to parallel chains through cytine within each polypeptide shown in figure 5. The bond is not broken under the unusual conditions of denaturation. Performic acid is used to oxidize insulin to separate the protein molecule into its constituents polypeptide chain without affecting the other part of the molecule. As shown in figure 5, two peptide chains joined by a disulfide bonds. Weak bonds, hydrogen bonds. The hydrogen bond appears from sharing of hydrogen atom between the nitrogen and the carbonyl oxygen of different peptide bonds is shown by the star. Each hydrogen bond is quite weak as shown in figure. Hydrophobic bonds, the non-polar side chains of neutral amino acids are closely associated with another proteins. No true bond exists. Nonetheless, hydrophobic bonds play an important role in maintaining protein structure. Now we discuss the structure of proteins. For a large macromolecule such as proteins, the task of describing and understanding the structure is approached at several levels of complexity, arranged in a kind of conceptual hierarchy. Four levels of protein structures are commonly defined as shown in figure 6. A description of all covalent and mainly peptide bonds and disulfide bonds linking amino acid residing in a polypeptide chain is its primary structure. The most important element of primary structure is the sequence of amino acids residue. Secondary structure refers to particularly stable arrangement of amino acid residue give rise to Recurring structural proteins. Tertiary structure of describes all the aspects of three-dimensional folding of a polypeptide. When a protein has two or more polypeptide subunits, the arrangement in space is referred to as quaternary structure. First of all, we discuss the primary structure. The peptide bond is formed by the amino acid linked by carboxyl group of one amino acid with the alpha amino group of another amino acid as shown in, for example, glycine. Thus, they form peptide bonds by several amino acids as alanine, glycine, histidine, leucine, etc. The primary structure of is ultimately becomes as shown in figure 7. It is the sequence of insulin, a glucose regulating hormones. Difference in amino acids at various positions of the two chains are few when comparing such diverse species as a horse or elephant. The similar structure shows a similarity in biological function so that the red insulins can be used by human beings. Then we can discuss as shown in the figure 7 secondary structure of secondary structure. Globular proteins have a coiled structure in which the peptide bonds are folded in regular manner as shown in figure 8. The folding are the results of linking of the carboxyl and amino group of the peptide chains by means of hydrogen bond and disulfide bonds. Such foldings are referred to as secondary structure of proteins. Present evidence is that in several proteins the hydrogen bonding produces a regular coiled arrangement called alpha helix. In alpha helix, Pauling and Corey were aware of the importance of hydrogen bonds in orienting polar chemical groups such as carbonyl and NH groups of the peptide bonds. They also had the experiments results by William Surrey in 1930 had conducting pioneering X-ray studies of proteins. Surrey shows that the proteins that make up hair and porcupine coils like this. Then a peptide chain forms regular helical coil called alpha helix. These coils are stabilized by hydrogen bonds between carbonyl of first amino acid and amide and form fourth amino acid residues. Thus, in a alpha helix, till a chain of hydrogen bonding is present. The alpha helix is shown, as shown in figure 9. According to Pauling and Corey, can be either right-handed or left-handed. Left-handed alpha helix is less stable than right-handed alpha helix because of steric interference between the carbonyl and the side chains. Only the right-hand alpha helix has been found in protein structure. In this structure, the polypeptide backbone is tightly wound around an imaginary axis drawn longitudinally through the middle of the helix, and the R group of the amino acid residue protrude outward form the helical backbone. The repeating units in a single turn of the alpha helix, which extends about 5.4 angstrom along the long axis, slightly greater than the periodicity. So, the amino acid residue in an alpha helix have conformation with alpha minus 45 degree to minus 50 degree and phi minus 60 degree, and its helical turns around include 3.6 amino acid residues. The helical twist of alpha helix found in proteins is right-handed. The alpha helix proved to the predominant structure of alpha kerateins. More generally, about one-fourth of alpha amino acids residue in polypeptides and found in alpha helix, the exact fraction varying greatly from one protein to the next. So, alpha helix forms readily because it makes optimal use of internal hydrogen bonds. The structure is stabilized by a hydrogen bond between the hydrogen atom attached to the electronegative nitrogen atom of a peptide linkage and the electronegative carbonyl oxygen atom of the fourth amino acid on the amino terminal side of the peptide bond. Within the alpha helix, every peptide bond participates in such hydrogen bonding. Each successive turns of the alpha helix is held to adjacent turns by 3 to 4 hydrogen bonds. All the hydrogen bonds combined give the entire helical structure considerable stability, as shown in this figure 5, figure 8 alpha helix structure. Thank you all of you.